Good morning, my fellow scientists. It's Thursday, January 19th, and science needs to get a little more caffeinated. Okay, that's better. Today I learned that the pangolin, or tank badger, has a tongue that is longer than its head and body, and it has the longest tongue-to-body ratio of any mammal. I bet it's very popular at the parties. In Allen Lab news, I am trying desperately to find a conformation switching aptamer selection scheme. So if you know of such a thing, please leave me a comment, send me an email, or a carrier pigeon. I don't care. I need a conformation switching aptamer scheme. What is a conformation switching aptamer scheme, you say? Well, buckle up, because I'm about to tell you. So an aptamer is a single-stranded nucleic acid, DNA or RNA, that binds to a particular target. The way you get an aptamer is you force DNA to evolve in a test tube to bind some particular target. After you've recovered the DNA that binds that particular target, sometimes some of those aptamers turned out that they fold up in a way that's detectable upon binding the target. Other ones don't change that much. So I need a selection scheme that deliberately searches for those particular aptamers that fold up very differently in their conformation between when they're unbound and when they're bound. Let me draw that for you. So we have this pool of lots of DNA molecules, each one is unique, and we add a target that we want to bind that's immobilized on some particle. And all these molecules sort themselves into ones that bind, like this one, and ones that don't. Now we can take the ones that bind and recover them using just a separation like magnetic particles or centrifugation, and we throw away all these waste DNA that don't bind, and we keep the ones that do. We elute them and amplify them, and we end up with a new pool of DNA. And you complete this whole process multiple rounds, maybe 10 or 20 times, and what you end up with is a set of DNA, all of which bind the target. Now that's all great, but there's no discrimination in this system between DNA that binds by folding around the target and DNA that just has the right shape already and binds the target without changing. So we want to select the ones that change, and we want to discard the ones that don't. How do we do that? So one of the ways that's been explored in the literature is to have your pool pre-bound to a particle, and then add your target, and pick out the DNA that is released upon binding the target. And that selection scheme has been published 2 or 3 times, and it seems to work, but I'm looking for something better. Tune in tomorrow for more about aptamers, research, DNA, and things with large tongues.